So let's talk first about how we preserve native structure within the microscope. And this is the subject of sample preparation. And I'm going to introduce you briefly to 4 different techniques for sample preparation. The first is traditional thin section EM. The second is staining and shadowing. The third is high pressure freezing. And the fourth is plunge freezing. So let's begin with traditional thin section EM. I call this traditional EM because it's the first set of methods that were developed historically to preserve the structure of a cell inside an electron microscope. These methods begin with chemical fixation using aldehydes, for instance. Now, the most common aldehydes are glutaraldehyde or formaldehyde. These molecules are cross-linkers. A cross-linker is a small molecule with 2 highly reactive groups on either side. And these reactive groups will form bonds with a variety of very common chemical moieties that are present on biological macromolecules. And so by adding these cross-linkers, they infiltrate inside the cell and they'll bind to almost anything that's next to something else. So for instance, they might stick to one protein and then bind to another protein right next to it. Or they might bind across 2 lipids in a membrane. Or they might bind a protein to a lipid membrane that's right next to it. And so the cross-linkers are almost like staples that just staple all the biological macromolecules to whatever is near it inside the cell. Another chemical fixative that's used is osmium. Osmium is a metal, but it's been found that infiltrating the cells with osmium causes the structure to become more rigid and be better maintained. Now, once the cell is chemically fixed, it can be dehydrated. Now, this is typically done by starting with the cell in probably an aqueous solution, washing it with a solution that may only be 90% water and say 10% alcohol. And then after that's completely washed through the sample, washing it again with a solution that's only 80% water and 20% alcohol. And then 70 and 30, eventually 50-50 water and alcohol. And then more alcohol than water until eventually the cells are bathed completely in some kind of alcohol or other organic solvent. To embed a cell in plastic, we first infiltrate the cell with a resin. For instance, a common popular resin is called EPON. And once the cell is filled with this resin, we then cure the resin to cause the individual monomers to polymerize and form a hard plastic. So curing is often done by raising the temperature well above room temperature, even in an oven. But there are also special resins that will polymerize at lower temperatures, which has the chance of preserving more of the cell structure. Once the resin is cured, it's hard, like a hard piece of plastic, and it can then be sectioned with a microtome. Once you cut thin sections through that plastic embedded cell, we then typically stain the sections with heavy metal stains. For instance, uranyl acetate, lead citrate are common stains. And each of these metals has a different affinity for different kinds of macromolecules inside the cell. For instance, uranyl acetate tends to decorate the protein components of the cell, while osmium tends to collect inside the membranes. And because the metals are coating the proteins and the lipids and the other macromolecules of interest, we can then image those metals because they have high contrast. Now, the sectioning step is done with an ultramicrotome, and I'm about to show a movie of how an ultramicrotome works. And what you'll see is that this is a chuck, and the sample is mounted at the very tip of the chuck. And then this blue object is a diamond knife. Actually, the knife blade is right here at the edge. And what you'll see is that the chuck moves up and down, and every time it comes down, it moves forward a little bit so that a little bit more will be cut off by the diamond blade, and then it moves forward and more is cut out by the diamond blade. And as sections are cut off onto the diamond blade, they float off into water, which is present in this little reservoir on the top of the diamond knife. So let's play the movie. So here we see the diamond knife being brought to the sample, and now the chuck moves up and down, each time pushing forward a little bit further over the diamond knife. Now if we look more close up from the top, we can see the tiny sections coming off of the block and floating off onto the water bath above the diamond knife. Now, while some of these procedures may have sounded harsh, they have nevertheless been marvelously successful. The traditional EM thin section strategies have filled our cell biology and histology textbooks with beautiful pictures of the interiors of cells. In fact, much of what we know about the interior of cells has come from this thin section EM. And as one example, I'll show this picture from a textbook on electron microscopy by Bozola and Russells. And in this picture is an image from traditional thin section EM of mouse urethra epithelium. In this image, this region is the lumen of the urethra, and these are the cells that line it. So this is the boundary of one cell, for instance. So here's one cell, and then here's the next cell next to it, and you can clearly see the membranes around it. Here's the boundaries of the next cell along in the epithelium. And so if you focus on just one cell, what are the features that we can see? Well, this image is highlighting the tight junctions that exist in between each of these cells. In addition, of course, you can see the nucleus. So here's the outline of the nucleus of this cell. This is the nucleus. And inside the nucleus, you can see heterochromatin as well as euchromatin in the middle. You can see mitochondria. Here's one, here's another, here's a third, and even the cristae inside. You can see spherical vessels as well as small tubular vesicles. So there's a lot of detail visible. This second example is one of my favorite traditional thin section EM images, and it's of insect sperm flagella. Again, it's in the book Electron Microscopy by Bozola and Russells. And what you see is packed insect sperm flagella. So this is the flagellum of one sperm cell, and it's packed right up against the flagellum from another sperm cell and another one, and they pack inside this tissue almost in a crystalline fashion. So each one of these is its own cell. Now inside the flagellum, the long skinny tail of the sperm, here is a mitochondria that will feed the flagellum energy, and then the basic structure of the flagellum is this series of microtubules. That's a microtubule, this is a microtubule, that's a microtubule. And so there's this pattern of 9 microtubules around the outside plus 2 microtubules in the middle in this flagellum. And if you look carefully at these microtubules, you can even see that you can see 13 protofilaments around the outside of the microtubule. While it's amazing that we can see even the individual protofilaments of a microtubule in this kind of thin section EM image, nevertheless, there are higher details that we can't get from this technique. For instance, we might wonder if we couldn't just zoom in with higher magnification on one of those protofilaments and see, for instance, where all the alpha helices are in the alpha-tubulin protein. Because electron microscopes can go to much higher magnification than is seen here. For instance, what if we tune it to 100,000 times magnification, or maybe 1,000,000, or even 5 million times magnification? Then could we see the alpha helices in the alpha-tubulin protein? The answer is no for a number of reasons. First of all, the alpha helices may not even be there anymore. Remember, this was a tissue that was first chemically cross-linked, and then it was dehydrated, and it's unclear whether the structure of the protein will remain intact when the water is removed away from it and it's placed in an organic solvent. After it was dehydrated, it was then plastic embedded, which is a very non-native condition. It was sectioned and then stained with a heavy metal. So it's unclear whether the alpha helices would have made it through that procedure in the first place. But even if they did, if we were to crank up the magnification and illuminate it with enough electrons to see detail, those incident electrons would destroy the helices long before we could ever resolve them. Instead, we could crank up the magnification and see the stain, the metal atoms that are surrounding that protein structure. And we could see the metals at higher and higher magnification because they can sustain a lot heavier dose than proteins. But we wouldn't be able to resolve the internal details of the protein themselves. Now let's move on to the next sample preparation technique of staining or shadowing. In this example of metal shadowing, purified RNA polymerase molecules were mixed with a solution of DNA. The RNA polymerase stuck to the DNA, as seen here. They're the dark spots on the DNA strand. And this solution was then dried onto a mica sheet. Mica sheets are very flat, and so the DNA is dried down with the RNA polymerases stuck to them. Then there was a metal shadowed onto that target. In this case, it was carbon and platinum. And the way this is sometimes done is you put the object of interest on a rotating plate, like an old record player, and as it's rotating in a vacuum chamber, you then put a rod of the metal that's going to be used for the shadowing, and you send a current through that rod, and as it heats up, clumps of metal atoms will come spewing off of the tip of the rod, and they rain down on the sample. The sample is rotating. So as a DNA strand comes around, clusters of metal atoms in front of it will get caught by the DNA strand, and it'll build up into a mound or a crest in front of the DNA molecule. Meanwhile, behind that crest, no metal clusters will hit just behind it because it's rotating, sweeping across. And as it rotates around and more metal rains down, the mound of metal in front of the DNA molecule grows and the shadow becomes more distinct. And this technique can generate fantastic, beautiful images that show individual DNA molecules, and in this case, of course, the RNA polymerases bound to it. Let me illustrate this on the tablet. Imagine that this is the grid, and then there's some object of interest on that grid, for instance, the DNA and the enzymes bound to it sitting on that grid. Now it's a rotating platform that's rotating around and around. So this is moving forward here, around and around. Now imagine that there's some source of clumps of metal that is raining down on the plate. And as it rains down, it produces an even coating in front of the object, but then more of that metal coating is collected on the front side of the object of interest, and then there's a little shadow behind it where the metal that would have fallen here is actually caught on the front face of this object because it's moving forward, sweeping forward. Then the next round, this will happen again, and so it'll get a little deeper all over the grid, but especially deeper right in front of our object. And as this gets deeper and deeper, the shadow that it casts will get longer and longer. So then another layer might come on like this, and then there's a very large amount of metal that will build up in front of the object, and that'll cast a very long shadow, so the third coating only goes like that. And as a result, you have a large amount of extra metal built up on the front side of that object, and it is the metal that would otherwise have fallen in this region, but it was captured on the front side. So this metal moves to there, and as a result, you have a very strong contrast. If you were to plot the density of the metal here, it would be at this level, and then it would drop and go almost to nothing, and then it would be very high here and then back to the baseline. And it's that contrast that you see in shadowing. The next example that I'd like to show is seen here, and this is a picture of a similar preparation technique, but the line going down the middle, like the trunk of the tree, is a ribosomal RNA gene. And in this cell, the cell was making ribosomes so quickly that many RNA polymerases were bound to that ribosomal RNA gene, and they were transcribing simultaneously. And here at the top, these RNA polymerases have just started, and so their transcripts are short. And as you go down further and further downstream in the gene, the transcripts get longer and longer, creating like longer limbs to the tree. And eventually, by the end of the gene, the transcripts are very long indeed here, and even some assembly is starting to happen at the bottom of the genes. So this just illustrates that metal shadowing can give spectacular molecular detail. But again, what you see is the contrast of the metal, and so you can't just crank up the magnification and look more carefully at the RNA polymerases, for instance, and see exactly what is the structure of RNA polymerase, because all that's preserved here is the replica of those proteins and DNA and RNA, in this case, that was built up as they were shadowed. Another form of shadowing is just simply called staining. In negative staining, let's say that is our grid, and that there are a couple objects of interest on that grid, maybe purified proteins, for instance, lying down on that grid. They're in a solution that has a lot of metal ions in it, say uranium, for instance, if this solution is uranyl acetate. And so the uranium is dissolved in this stained solution. And as that solution evaporates, the metal ions are concentrated to higher and higher concentration in the thinning layer of fluid until eventually they precipitate. And as they precipitate, they form a film here along the grid, and the film gets deeper and deeper and surrounds the proteins. And ideally, for instance, for a short protein, the film of negative stain would completely cover the protein. What's seen with larger proteins or complexes is that often a little meniscus forms around the protein where the stain is extra deep, and then it has its standard thickness elsewhere. And so this is what the grid might look like after the stain dries. As a result, there will be very high contrast where the proteins were. The depth of scattering material in the background will be relatively constant, but then right around a protein, that depth will rise strongly and then drop immediately right around where the protein is, and so create a very high contrast object. Elsewhere, if a protein is buried in the stain, what you see is the absence of stain in that spot. You see basically a hole in the stain with maybe areas where the stain is deeper, and then it goes back into background. And so the stain tends to highlight the particles. And this is an example image that was obtained by negative staining. In this case, it's a purified protein, the 20S proteasome. Over here is the crystal structure, a representation of the crystal structure of the 20S proteasome from the side view, where you can clearly see that it's barrel-shaped. And then down here, a top view down the barrel. Now, when this protein was applied to an EM grid and stained with uranyl acetate, you can see that the stain clearly reveals the barrel shape of the protein, because the stain collects around the edges of the protein and makes those edges distinct. So for instance, right here is the side view of a barrel-shaped 20S proteasome. And then down here, for instance, is a top view of a 20S proteasome. So negative staining can show you the shape of individual protein molecules.